Pepsin is an enzyme that breaks down proteins into smaller peptides. It is produced in the stomach and is one of the main digestive enzymes in the digestive systems of humans and many other animals, where it helps digest the proteins in food. It is one of three principal proteases in the human digestive system, the other two being chymotrypsin and trypsin. During the process of digestion, these enzymes, each of which is specialized in severing links between particular types of amino acids, collaborate to break down dietary proteins into the components i.e. peptides and amino acids, which can be readily absorbed by the small intestine. Pepsin is most efficient in cleaving peptide bonds between hydrophobic and preferably aromatic amino acids such as phenylalanine, tryptophan, and tyrosine. Pepsin's proenzyme, pepsinogen, is released by the chief cells in the stomach wall, and upon mixing with the hydrochloric acid of the gastric juice, pepsinogen activates to become pepsin. Pepsin is an aspartic protease, using a catalytic aspartate in its active site. History. Pepsin was one of the first enzymes to be discovered. It was discovered in 1836 by Theodor Schwann. Schwann coined its name from the Greek word pi epsilon psi iod sigma pepsis, meaning digestion. Scientists around this time began discovering many biochemical compounds that play a significant role in biological processes, and pepsin was one of them. An acidic substance that was able to convert nitrogen-based foods into water-soluble material was determined to be pepsin. In 1928, it became one of the first enzymes to be crystallized when John H. Northrop crystallized it using dialysis filtration and cooling. Precursor. Pepsin is expressed as a zymogen called pepsinogen, whose primary structure has an additional 44 amino acids. In the stomach, chief cells release pepsinogen. This zymogen is activated by hydrochloric acid, which is released from parietal cells in the stomach lining. The hormone gastrin and the vagus nerve trigger the release of both pepsinogen and hydrogen chloride from the stomach lining when food is ingested. Hydrochloric acid creates an acidic environment, which allows pepsinogen to unfold and cleave itself in an autocatalytic fashion, thereby generating pepsin. Pepsin cleaves the 44 amino acids from pepsinogen to create more pepsin. Activity and stability. Pepsin is most active in acidic environments between 37 degrees Celsius and 42 degrees Celsius. Accordingly, its primary site of synthesis and activity is in the stomach. Pepsin will digest up to 20% of ingested amide bonds by cleaving preferentially after the N-terminal of aromatic amino acids such as phenylalanine, tryptophan, and tyrosine. Pepsin exhibits preferential cleavage for hydrophobic, preferably aromatic, residues in P1 and P1 positions. Increased susceptibility to hydrolysis occurs if there is a sulfur-containing amino acid close to the peptide bond, which has an aromatic amino acid. Pepsin cleaves Phe1-val, GLN4 his Glu13 ALA, ALA14 LEU, LEU15 TIR, TIR16 LEU, Gli23 Phe, Phe24. Pepsin exhibits maximal activity at pH 2.0 and is inactive at pH 6.5 and above. However, pepsin is not fully denatured or irreversibly inactivated until pH 8.0. Therefore, pepsin in solution of up to pH 8.0 can be reactivated upon reacidification. The stability of pepsin at high pH has significant implications on disease attributed to laryngopharyngeal reflux. Pepsin remains in the larynx following a gastric reflux event. At the mean pH of the laryngopharynx, pepsin would be inactive but could be reactivated upon subsequent acid reflux events resulting in damage to local tissues. In laryngopharyngeal reflux, Pepsin is one of the primary causes of mucosal damage during laryngopharyngeal reflux. Pepsin remains in the larynx following a gastric reflux event. While enzymatically inactive in this environment, pepsin would remain stable and could be reactivated upon subsequent acid reflux events. Exposure of laryngeal mucosa to enzymatically active pepsin, but not irreversibly inactivated pepsin or acid, results in reduced expression of protective proteins and thereby increases laryngeal susceptibility to damage. Pepsin may also cause mucosal damage during weakly acidic and non-acid gastric reflux. Weak and non-acid reflux is correlated with reflux symptoms and mucosal injury. Under non-acid conditions, pepsin is internalized by cells of the upper airways such as the larynx and hypopharynx by a process known as receptor-mediated endocytosis. The receptor by which pepsin is endocytosed is currently unknown. Upon cellular uptake, pepsin is stored in intracellular vesicles of low pH at which its enzymatic activity would be restored. Pepsin is retained within the cell for up to 24 hours. Such exposure to pepsin at neutral pH and endocytosis of pepsin causes changes in gene expression associated with inflammation, which underlies signs and symptoms of reflux and tumor progression. This and other research implicates pepsin in carcinogenesis attributed to gastric reflux. Pepsin in airway specimens is considered to be a sensitive and specific marker for laryngopharyngeal reflux. Research to develop new pepsin-targeted therapeutic and diagnostic tools for gastric reflux is ongoing. A rapid non-invasive pepsin diagnostic called PeptiDS is now available, which determines the presence of pepsin in saliva samples. Storage. Pepsins should be stored at very low temperatures to prevent autolysis. Inhibitors. Pepsin may be inhibited by high pH or by inhibitor compounds. Pepstatin is a low molecular weight compound and potent inhibitor specific for acid proteases with a Ki of about 10-10 m for pepsin. The statal residue of pepstatin is thought to be responsible for pepstatin inhibition of pepsin. Statin is a potential analog of the transition state for catalysis by pepsin and other acid proteases. Pepstatin does not covalently bind pepsin, and inhibition of pepsin by pepstatin is therefore reversible. 1-bis-2-penylethane reversibly inactivates pepsin at pH 5, a reaction which is accelerated by the presence of Cu. Pepsin also undergoes feedback inhibition. A product of protein digestion slows down the reaction by inhibiting pepsin. Sucralfate also inhibits pepsin activity. Applications. Commercial pepsin is extracted from the glandular layer of hog stomachs. It is a component of rennet used to curdle milk during the manufacture of cheese. Pepsin is used for a variety of applications in food manufacturing, to modify and provide whipping qualities to soy protein and gelatin, to modify vegetable proteins for use in non-dairy snack items, to make pre-cooked cereals into instant hot cereals, and to prepare animal and vegetable protein hydrolysates for use in flavoring foods and beverages. It is used in the leather industry to remove hair and residual tissue from hides, and in the recovery of silver from discarded photographic films by digesting the gelatin layer that holds the silver. Pepsin was historically an additive of Beeman's gum brand chewing gum by Dr. Edward E. Beeman. It also gave name to Pepsi Cola, originally formulated with pepsin and cola nuts. Pepsin is commonly used in the preparation of F2 fragments from antibodies. In some assays, it is preferable to use only the antigen-binding portion of the antibody. For these applications, antibodies may be enzymatically digested to produce either an Fab or an F2 fragment of the antibody. To produce an F2 F2 fragment Ig is digested with pepsin, which cleaves the heavy chains near the hinge region. One or more of the disulfide bonds that join the heavy chains in the hinge region are preserved, so the two Fab regions of the antibody remain joined together, yielding a divalent molecule, hence the designation F2. The light chains remain intact and attached to the heavy chain. The Fc fragment is digested into small peptides. Fab fragments are generated by cleavage of Ig with papain instead of pepsin. Papain cleaves Ig above the hinge region containing the disulfide bonds that join the heavy chains, but below the site of the disulfide bond between the light chain and heavy chain. This generates two separate monovalent Fab fragments and an intact Fc fragment. The fragments can be purified by gel filtration, ion exchange, or affinity chromatography. Fab-own F2 antibody fragments are used in assay systems where the presence of the Fc region may cause problems, in tissues such as lymph nodes, the spleen, or in peripheral blood preparations. Cells with Fc receptors are present which can bind the Fc region of intact antibodies, causing background staining in areas that do not contain the target antigen. Use of F2 or Fab fragments ensures that the antibodies are binding to the antigen and not Fc receptors. These fragments may also be desirable for staining cell preparations in the presence of plasma, because they are not able to bind complement, which could lyse the cells. F2, and to a greater extent Fab fragments allow more exact localization of the target antigen, i.e. in staining tissue for electron microscopy. The divalency of the F2 fragment enables it to cross-link antigens, allowing use for precipitation assays, cellular aggregation via surface antigens, or rosetting assays. Genes. The following three genes encode identical human pepsinogen A enzymes. A fourth human gene encodes gastrixin, also known as pepsinogen C.